Just before I start, I want to acknowledge some of the folks that have done some of the work that I'll show you to you today. Usman Arshad is former PhD student. Juan Bolatti, a master's student. Marcus Zenobi, probably many of you know him. He works with Balchem. And my colleague, Charles Staples, passed away. So I'm going to share data with you. I'm going to be selfish. I'm going to show mostly data from University of Florida. It doesn't mean that's the best data out there or it's the only data out there. It's just for the sake of time. I cannot show you everything that there is on choline in ruminants. So just be aware of that. There's other work that's been done by Barry Bradford, Heather White, Richard Mann, many experiments here in Europe, in Italy, and the Netherlands. So I'm going to just show my side of the story, but just because of time. So I'll tell you a short history on choline, why it became important, particularly in human nutrition. Then I'll touch on the role of choline on intermediary metabolism. And then obviously the classic role of choline as a lipotropic agent to reduce hepatic triacylglycerol concentration. And then I'll get into the effects of choline supplementation after the development of rumen-protected products, particularly during the transition period. I want to just touch on this concept here because there's probably some misinformation in the field relative to what animals might respond to choline supplementation. And then hopefully by the end of my presentation, I'll make the case that choline fits this concept, not necessarily an essential nutrient like it is considered in the human literature, but a required nutrient that should be part of diets of ruminants. And obviously, it is part of diet of young calves, so choline chloride is supplemented in diets of young calves, but once that calf is weaned or it doesn't receive milk replacer, it's sort of forgotten. So this all started in the early 1700s, but it only became really important during this discovery of insulin in Canada by Frederick Banting, whom eventually went to win a Nobel Prize in Physiology and Medicine. So during the work to understand and characterize insulin, him and his student, Charles Bass, was a medical student at the University of Toronto. They realized that their animal model that they used to study insulin at the time or to discover insulin, they depancreatize these beagles, these animal models, these dogs. And Charles Bass observed that they would develop fatty liver. So they removed the pancreas of those dogs. They would develop eventually fatty liver, and many of them would succumb with fatty liver after the experiments were completed. So when Charles Bass, whom eventually became a member of the National Academies of Science in the United States, went on to a very illustrious scientific career, he conducted initial experiments showing that if they supplemented back to those dogs pancreas, and eventually they supplemented to those dogs phosphatidylcholine, and then eventually choline chloride, they were able to reverse some of those effects. So the degree of infiltration of triacylglycerol in the liver of those depancreatized dogs would be minimized. And you can see here, this was done almost 100 years ago, 90 years ago, yeah? So in the ruminant world, it was here in England that this work on choline initially started. And Derek Lindsay here was the first to use carbon-labeled choline to show that it was in sheep, it was almost all degraded by the rumen microbes, and all phosphatidylcholine pool in the ruminant plasma and in tissue came from de novo synthesis. Okay, so being synthesized from other substrates. Okay? This sort of stayed quiet for some time until Rich Erdman at the University of Maryland did the initial infusion work, at least that I'm aware of, with choline or choline chloride, either in the rumen or in the abomasum or in the duodenum, and really the initial work with rumen-protected choline. And then he confirmed that there's extensive microbial degradation. So more than 90% of the choline present in feeds, most likely in the form of phosphatidylcholines or lysophosphatidylcholines, was degraded by the rumen microbes. So very little actually showed up in the small intestine. So obviously because of that, it became difficult to study and still is difficult to study. And I think that's probably why there's some confusion out there, because we can't really quantify precisely how much gets there, how much gets absorbed. And choline is a complicated nutrient because it gets taken up by tissues during first pass in the intestine and converted in multiple metabolites. So here is a diagram of the main pathways by which choline is metabolized by tissue. So obviously choline is substrate for synthesis of neuromediators such as acetylcholine. So it can be acetylated in neurons to form acetylcholine, which is a neurotransmitter. So important for muscle contraction, nerve impulse propagation. So that's one of them. So if choline is consumed, it can be acetylated in neurons or in central nervous system. Or it can be phosphorylated through this pathway that's called Kennedy pathway, because it was characterized by a US biochemist, and he was given his name. And this takes place in probably every tissue we are aware of. So it's converted to phosphocholine, and then through a series of reactions, it eventually is used to synthesize numerous phosphatidylcholines depending upon the sequence of fatty acids, of the two fatty acids that are associated with the main backbone. So in ruminants, this is thought to be a minor pathway just because there's not a lot of choline being absorbed in the gastrointestinal tract. And then choline can be oxidized to betaine. And betaine is considered one of the important methyl donors. So it's part of this betaine, folate, methionine pathway that eventually can generate acetylmethionine, that's considered the universal donor of methyl groups. That's important to control, for instance, gene expression, how genes become silent or genes are expressed so proteins can be synthesized. So during this process, there is an interplay with this molecule that's important in the human medicine. It's a marker for coronary heart disease, homocysteine accumulation. So through the action of this enzyme here, betaine homocysteine methyltransferase, you can donate methyl groups to regenerate methionine from homocysteine. And this is enhanced by the presence of vitamin B compounds. So there's a pathway that's not here. It's called the transsulfuration pathway. So folic acid is important for this pathway, and then pyridoxine is important for the transsulfuration pathway. So then methionine can condense with adenosyl to form S-adenosyl methionine, and this becomes a donor of methyl groups that can methylate sites of the DNA, and in general, this silence gene expression, so proteins are not produced in a particular tissue. Okay? And this takes place by this interplay with phosphatidylethanolamine. So this happens at the level of the liver, to the best of my knowledge. That's the only tissue that does that. So if we were to think about ruminants, if this pathway is limited, then the ruminant becomes highly dependent on this pathway here of transmethylation for synthesis of phosphatidylcholines, which are the main phospholipids, and they are used for synthesis of every possible membrane, cell membrane, organelle membrane, in addition to many other functions in the body. So if you think of a dairy cow, that's the focus of today, obviously in early lactation, we have limitations in supply of amino acids. The same way we talk about dairy cow that's in negative energy balance, she's for some weeks in negative amino acid balance, negative mineral balance, whatever you think of that's happening those first few weeks of lactation. So this pathway is probably limited in dairy cows at the beginning of lactation just because the short supply of essential amino acids, in particular in most of our diets, short supply of methionine. Okay, so choline is required as a compound that's part of phosphatidylcholine, which is the main phospholipid. You know, it's part of sphingolipids, phospholipids that are important for the structure of cells, but also for the fluidity of cell membranes, for absorption and transport of nutrients in the body. It plays a role in this so-called one-carbon metabolism, which is this transfer of methyl groups that can alter how genes are expressed in particular tissues. Obviously, it's quite important for lipid and cholesterol transport. That's the case of fatty liver, for example, that I just mentioned to you. So if you think of Dr. Clay Zimmerman, he needs 500 milligrams a day of choline. Okay? In excess, choline is probably not very good for humans. Choline, carnitine, they're converted to these trimethyl amine oxides that today medical literature says it's linked to cardiovascular disease. But if you don't consume enough, then you develop issues related to choline deficiency, one of which is fatty liver. In the poultry industry, we have guidelines for feeding laying hens. We have guidelines for feeding growing chicks. We have guidelines for feeding lactating sows, but really don't have a specific value for lactating dairy cows. And I think the major issue is really related to the fact that we don't have dose response experiments to titrate the exact dose. Second, we don't have infusion experiments to precisely know how much got to the small intestine, which is difficult to do this with many animals. Third, different products have probably different bioavailability of choline. So it's just hard to pinpoint an exact number of how much of that molecule is needed by a cow. Okay? So because of that, guidelines that are put forth, such as the NASEN in the US, would tell you that choline is important, but it wouldn't give you the exact value. So in your software, there won't be a red flag because you didn't add choline to the diet of a lactating cow. Here, just to show that as many molecules, phosphatidylcholines are at smallest concentration right around parturition. Okay, so obviously this is being sucked up by the fetus for synthesis of tissue. It's being sucked up by the mammary gland for renewal of cells and synthesis of colostrum and milk fat globule. But also we perhaps have the limitation in supply of substrate for synthesis of phosphatidylcholine. In particular, probably limitation in supply of methionine at this stage. Okay? So this makes the case that perhaps choline is really important at that time in the lactation cycle. So what are some of the roles of choline at this stage? So if you think of your typical cow, right close to calving, there's this exponential growth of the pregnant uterus, in particular the fetus within the pregnant uterus. Most cows in the last few days of lactation will drop dry matter intake. So if you look individually, not every cow, but most of them will drop dry matter intake in the last 2 or 3 days of gestation. Okay, so intake is less, at the same time the nutrient needs increase. And obviously there's all the adaptation to lactation that shifts metabolism towards either the fetus or the mammary gland, so the cow can adapt to that. And as a consequence, she mobilized tissue. It's a perfectly normal process in any pregnant mammal or early lactation mammal. Okay? But in the cow in particular, when we have excessive, particularly excessive amounts of adipose tissue, the concentrations of free fatty acids in blood will increase. And obviously, there is a supply through the arterial supply to tissues, It can be taken up by the mammary gland for synthesis of long-chain fatty acids just by incorporation to milk fat. It can be taken up by muscle tissue, but it can also be taken up by the liver. Obviously, the liver has a certain amount of ATP that's needed. Once you meet the needs of energy by those cells, it will divert pathways as opposed to completely oxidizing to incomplete oxidation. So you preserve nutrients. So you synthesize ketones. That's somewhat misunderstood in the industry because most particularly in the veterinary profession think of this as being a bad thing. And this is a way of surviving starvation. So it's actually a quite elegant pathway to preserve nutrients. So increasing ketones is just an indication of excessive tissue mobilization, fatty tissue mobilization, but it's not necessarily a disease per se. Okay? So these ketones can now be exported. They are water soluble. They don't require protein to be transported. And this will be used as a source of energy, such as for de novo synthesis of fatty acids, so short, medium chain fatty acids in the mammary gland. But obviously, when in excess, that can lead to re-esterification of fatty acid in the hepatocytes. And that will accumulate these lipid droplets, which are quite dynamic in the liver. And this might lead some cows to develop fatty liver, which is linked to negative consequence during lactation. Okay? Either because fatty liver causes alterations in the hepatocyte, or just because it's associated with an animal that doesn't eat well. Okay? Now, ruminant liver was never designed to export a lot of triacylglycerol, just because of the type of nutrients they absorb, which is primarily based on short-chain fatty acids. So the liver is not really an adipose or a fatty acid synthesizing type of tissue like the mammary gland is or the adipose tissue is, as opposed to our liver that is designed to handle a lot of glucose that gets absorbed. In ruminants, that's really not the case. So because of that, ruminants never had to export a lot of triacylglycerol. So there is a slow process of eliminating the excess of triacylglycerol that's accumulated, because the ability to export is limited through lipoproteins, and it relies primarily on oxidative pathways. So when in excess, that can damage the hepatic cell. It can cause a pro-inflammatory response. It can induce acute phase protein synthesis. So if somehow we could twist the liver to export a little more as a very low density lipoprotein, perhaps we could enhance the ability to deal with fatty liver at some stages of lactation. I'll come back to this during my presentation. So at this time, I think it's fair to assume that the supply of essential amino acids or amino acids in general is less than adequate. So methionine is limited. We know that choline is degraded. So it creates this story that it might be an optimum time for cows to respond to rumen-protected choline. So some cows will develop things like this, the Marcus-induced fatty liver in cows, and this is what he got. Needless to say that she didn't survive, yeah. So there's a lot of triacylglycerol accumulating in that liver, and the liver is gigantic. So typical bovine liver weighs 11 kilos, 12. This is probably in the 18 kilograms to 20 kilograms in this cow. So when do they develop fat accumulation in the liver? Right at calving. So these are data from multiple published experiments. Each dot represents a treatment group. So Marcus just pulled this out of literature. Some studies sample liver tissue multiple times, but you can see there is a pattern. As cows begin lactation, there's an accumulation of triacylglycerol in the wet tissue of the liver. So liver is about 35 to 40% dry matter. The remainder is water. So on a dry matter basis, obviously, this would be 3 times greater. So it would be 20-some% to 30% of the tissue is triacylglycerol. Okay? So typical liver composition of a well-fed cow contains less than 3% triacylglycerol on a wet basis. So typically 1.5 to 2%. So as this triacylglycerol increases, glycogen decrease because of glycogenolysis. And this sort of sets the stage for cows to develop hepatic steatosis. So how common is that? It's actually quite common in literature. So multiple studies or experiments have been done. So this is in England, this is in the Netherlands, the remainder are in the US, have characterized liver composition. The liver is not a completely homogeneous tissue, I should say, but you can see that about half of the cows have at least a moderate degree of hepatic steatosis, and perhaps 10 to 20% have really excessive, severe triacylglycerol. And this would be linked to metabolic crisis and failures, those cows that people treat, and they don't get better. Okay, so I think it's fair to assume that a large proportion of cows have hepatic steatosis. So there's an association between hepatic steatosis and how the cows perform in that lactation. Whether that's a causal relationship, we don't know because we don't have these types of experiments that we induce lots of cows to develop fatty liver. But here's data from literature in which we pull our own experiments that we collect liver tissue, and we basically interrogate the question, is there an association between hepatic triacylglycerol concentration in the first week after calving, so it's actually average of day 8, and subsequent survival, production performance, and reproduction. So I'll show you just, I think just this slide here, where you can see that we use hepatic triacylglycerol as a linear or a continuous covariate in our statistical models. Okay, but just for the ease of interpretation, I'm illustrating what would happen if in our data set, cows would go from 2.5 to 7.5% triacylglycerol. You can see the risk of metritis, which is the cow that has the watery, reddish, brownish, fetid type of discharge in the first 2 weeks of lactation. That makes them look very sick. Doubles. The risk of metritis associated with fever or sepsis also doubles. The risk of morbidity increases substantially. Cows that have more than one clinical disease increases substantially. So they have a lot of other problems associated. So we like to think that fatty liver complicates the life of the cow even more, and that predisposes them to other disease. So where does choline get into this? So this is probably one of the first, if not the first experiment with dairy cows in which choline was, or room-protected choline was shown to reduce hepatic triacylglycerol concentration. So this was done by Dr. Rick Grammer in the early 2000s. One of his visiting students from Brazil, Reinaldo Cook, did this experiment. So they had a model, a feed restriction model, to induce hepatic steatosis in late gestation dry cows. So they basically underfed those cows to develop fatty liver, okay? And they had cows that were fed a controlled treatment or a room-protected choline supplemented treatment. So these cows were being fed 3 to 4 kilograms of dry matter a day. So you can realize that they're going to mobilize a lot of body tissue, including triacylglycerol from fat depots, okay? So what they showed was that in the pre-treatment, the composition of the liver relative to triacylglycerol did not differ between the two treatments. But after they were induced to develop hepatic steatosis, almost every cow developed fatty liver, but the degree of hepatic steatosis was less when they received room-protected choline. So that's the first indication that the cow is not different from other animals, and they actually respond to choline by reducing hepatic infiltration triacylglycerol. Then Marcus Zenobi, during his experiment, during his PhD, conducted an experiment in which we showed that hepatic triacylglycerol decreased linearly as the intake of choline ion as choline chloride that was encapsulated. So choline chloride is about 75% choline on the molecular weight. So, so this here, 12.5 grams of choline ion would be your typical recommended dose by Balchem. But you can see that that's not the ideal dose to reduce triacylglycerol. So you can see during the ad libitum feeding, no difference. It doesn't really matter whether you fed choline or not. Hepatic triacylglycerol was the same. These were well-fed cows. But when we induce hepatic steatosis, it decreased linearly as the intake of choline ion increased. So another evidence that it has this lipotropic effect. So a few years ago, we replicate this experiment with a small twist in the design. So we assign 110 late pregnant dry hosting cows to this fatty liver induction protocol. Okay? And then we assign them to one of 5 treatments. The treatments were our negative control, 0 grams of choline ion, exact same diet, and exact same protocols, except that they didn't receive any choline ion, they just received the encapsulation material, the lipids that are used to encapsulate choline. And then we fed 2 products at 2 levels. I want to focus on the amount and not necessarily on the product. You guys can ask questions to Clay later on about the difference in products, but I'm more interested in going from 0 to 12.9 to 25.8 grams of choline ion. Okay, so let's just keep that in mind. I'm not going to touch on whether we fed a less concentrated or a more concentrated product, because you will see that there's only minor differences, almost none, to be honest. So we had a period of 5 days of leptin intake, followed by 9 days of feed restriction. And these cows were restricted to 42% of that net energy for lactation required at that stage for maintenance and pregnancy. So to make a long story short, these cows were eating about 5 kilograms of dry matter for 9 days on average. So they mobilized tissue. So here is, we collect liver tissue on day 6 of the experiment when they were still being fed ad libitum, and then on day 13. So our comparisons here will be the orthogonal comparison is the average of choline supplementation against the control. And then we have a factorial arrangement here, but I'm going to focus on the 25.8 versus the 12.9. So that's what I'm interested in. So here's the liver triacylglycerol content on an as-is basis or adjust for the dry matter content. So you can see here the superscripts, what they mean. So here's the asterisk is the effect of choline. And then I'm interested in the effect of choline and the effect of the amount of choline. Okay? So you can clearly see that choline reduced triacylglycerol, whether we express that on as-is basis or dry matter basis. And as we fed more, it reduced even further. Okay? So as we increase the dose, it further reduced triacylglycerol. So if we look at glycogen, it's exactly the opposite response. And that is a repeatable response that we don't necessarily understand very clearly how this is happening. There's some hypotheses out there. But when cows are induced to develop a fatty liver, or if bovine hepatocytes are cultured in vitro in presence of choline chloride, there's an increase in glycogen content. And we don't know if it's because of shifts of how carbon gets used by the cell, or is the effect of accumulation of water that typically beta induces within the cell. It's an osmotic regulator of cells. And at least based on the rat literature, when cells accumulate a little bit more water and they are heavier, that induces glycogen synthesis, glycogenesis in the cell. Okay? So this creates a phenotype of the liver tissue that is less conducive to fatty liver, which is a smaller ratio of triacylglycerol to glycogen. So typically when you have fatty liver, the ratio of triacylglycerol to glycogen goes way up because there is more fat accumulated in the liver, and the cow mobilizes glycogen to produce glucose through glycogenolysis. So favoring, let's say, quote unquote, liver health, yeah? So what happened to blood metabolites? So in the well-fed state, concentrations were less. When we induce fatty liver and we starve cows, it goes up. But choline does not change that. And I think this is an important message for people in the field. I've been to farms in which people want to measure the success of supplementing choline by measuring BHB. You should not expect reduction in ketogenesis. Do not use that as your gauge to tell whether your rumen-protected choline is working or not. I think that's a mistake and it should not be done. Okay? But there is less inflammatory response. So when we saw this data here, actually I told the student, you messed up with the assay. We do randomized block design, so we do all the analysis based on the block that we randomized out to the experiment. I said, this cannot be possible. So he went back to the lab and he repeat all the 110 cows and all the time points, and that's exactly what happened. So we replicate that in a different experiment. So there is this reduction in acute phase response. We don't think it's caused by choline per se, but it's probably a combination of either reduced triacylglycerol in the liver, so less fatty liver, so there's less insult and inflammation, but also because of increase in the pool of phosphatidylcholines that have anti-inflammatory effects. Okay? So now, here's data from cows from epidemiological studies in which they had cows that they call the so-called control, so no fatty liver, or they had cows that had fatty liver. And this group here measured some key genes and protein in the liver tissue that are important for assembly and secretion of very low density lipoprotein. This microsomal transfer protein, this apolipoprotein B100, and this apolipoprotein E are all required either as constituents of the VLDL or for transfer of triacylglycerol among organelles so it can repackage as a lipoprotein. And one of the things they showed was that this is protein, so it's actually better than gene expression, is that cows that develop fatty liver have less protein expression for those three important compounds. Whether that caused the fatty liver or is a consequence of fatty liver, we cannot sort that from this study. But there are other consequences for inadequate choline supply. So this is data in rats, and this is a different type of polypoprotein. This is synthesized by enterocytes. It's called a polyprotein B48 because it's truncated at a particular codon, so the translation of the mRNA stops, so it manufactures a different protein. So this is typical of what's called chylomicrons that help absorb lipids from the gastrointestinal tract into the lymphatic system of the animal so it can be dispersed across the body. And what they showed was that when they fed a choline deficient diet, the amount or concentration of polypo B48 was less, suggesting that there is less synthesis of lipoproteins in the intestine, chylomicrons, therefore less absorption of lipids in the gastrointestinal tract of laboratory animals. So this builds a story that, yeah, we know choline is important for synthesis of phospholipids, which are integral part of lipoproteins that have multiple uses. So given what I told you, we asked the question, is that also true in ruminants? And we use an indirect approach to measure that. I wish I had the skills, Dr. Chris Reynolds, to do the multi-catheterized type of experiment so we could look at flux of lipoprotein from the liver. But we use a technique that is used in laboratory animals, that is this non-ionic detergent that coats the lipoprotein particles. Okay? So what we did is we induced cows to have fatty liver, and we, to the best of our knowledge, based on data with guinea pigs and mice, we used this dose here of thiloxapol. Okay? So this induces hyperlipidemia in cows. And what it does, this is a diagram or a picture of a VLDL particle. So this VLDL is secreted by the liver as nascent VLDL. And then lipoprotein lipases that are part of blood vessels, or they are in tissues, hydrolyze the core of the VLDL to hydrolyze the triacylglycerol and release fatty acids so tissues can take up. Think of lipids that are being transported in the blood, and the mammary gland wants to suck up some of that to put into milk fat. Or your muscle wants to use some of that as energy source. Okay? Particularly in early lactation, that maybe the supply of short-chain fatty acids, acetate in particular, may not be ideal. So when we use thiloxapol, it coats the VLDL particles. So lipoprotein lipase is unable to hydrolyze that. So then it accumulates over time. And you can use that as a proxy for the rate of secretion by the hepatocyte in the live animal. Okay? So this is what we've done here. But we had 3 treatments. But really I'm interested in 2 of them, which is the mean effect of the 25.8 grams of choline ion, either with one of the 2 products, which I'm not going to touch on that, versus the 0 grams, the control. So we had 33 cows, 11 cows per treatment here, but we really have the mean of 22 cows against the mean of 11 cows. So we collect lots of things, liver tissue, lymph from the supramammary lymph vessels. Okay? And we were interested in secretion of triacylglycerol by the liver. So this is what we found. So as time goes by, triacylglycerol in blood increases. These cows were off feed for 48 hours, and they were fed a diet that almost had no lipid at all. So all of this triacylglycerol is coming primarily from secretion by the liver. So it's VLDL, nascent VLDL triacylglycerol. And what we found was that the rate of secretion was greater in the dashed lines, which represents the cows that receive choline ion as room-protected choline. So if you want to see this better, the area under the curve was greater for the cows supplemented with choline than the control cows. So corroborating the data that we knew for 90 years now that it increases VLDL secretion. So cow doesn't seem to be a lot different than other animals, although the rate of secretion is probably a lot less than in monogastrics. Okay, so we look at a lot of gene expression to look at these molecular signatures of the tissues when we feed or don't feed choline or associated with hepatic triacylglycerol. So I'll show you some of that and then I'll give you a general summary. So this is choline kinase. So the way this is expressed is a little odd. It's just because the way gene expression is quantified in laboratory. So here we use the negative value, okay? So, but hopefully you trust me. So as the number goes down, the expression of that gene decreases. So you can see there is an association between how much triacylglycerol is in the liver and the expression of this particular gene here. This is the gene that activates choline to become phosphocholine in that initial Kennedy pathway to synthesize phosphatidylcholines. Okay, so another gene here that's involved in the synthesis of phosphatidylcholine, you can see there's a negative relationship. Okay, so as I have more fatty liver, the expression of that gene goes down. We can't really from this data sort whether it's the reduced expression that induces more fatty liver or the other way around. So that's just not possible here. Now let's look at genes that are important for assembly and secretion of lipoprotein. So here is the ApoB100, the big apoprotein that's part of VLDL. We have the same pattern here. So more fatty liver, less expression, at least of mRNA for a polypoprotein B100. The same thing, that's the microsomal transfer triglyceride protein. The level of expression goes down. So all of these genes involved in assembly and secretion of lipoproteins by the liver are associated with fatty liver. So it seems that the data that I showed initially with the protein level replicates the gene level here. So we think there's probably some causal relationship as well. So in a subsequent experiment, we asked the question, does supplement choline, because choline is also important for synthesis of chylomicrons, which are important for lipid transport from the gastrointestinal tract. Does that change how fat gets absorbed in the intestinal tract? So we induce, we feed restricted cows, okay? And then on day 9 of the experiment, we dosed them with 400 grams of long-chain fatty acids, primarily palmitic acid, plus 450 grams of cornmeal. So these cows were off feed. And then, at least on this, just prior to that. On this day here, we took blood samples, we took supramammary lymph samples, and we took fecal samples sequentially to use indigestible NDF, a 288-hour undigested NDF as an internal marker to look at digestibility of fat in those cows. And then we measure a few things in those samples. So here is the serum triacylglycerol. So again, I'm interested in the mean of these two against the control. You can see the dashed lines have greater concentration of triacylglycerol. They had greater concentration of choline in the supramammary lymph content. Although that's not the lymph that's drained in the intestine, we make some assumptions here. So we think that there is greater secretion of chylomicrons, although I cannot prove that to you. But we did increase total tract digestibility of fat. So from about 80% to about 85%, so about 5 percentage points. So we think that this story is important because that enhances and it makes sense. Now, is the mechanism linked to chylomicrons, or perhaps the mechanism is linked to the integrity of the architecture of the intestine? We don't know, because there's data in literature showing that if you starve animals from choline, you disrupt tight junctions in the intestine, and that alters the ability of absorption of the intestine in laboratory animals. Because we underfed these cows in this experiment, and there's good data showing if you underfeed cows to mimic what happens in the day of calving, for example, the architecture of the intestine is disrupted, tight junctions become weaker, so things flow across through paracellular mechanisms. So it's possible that choline, by its effects on the synthesis of phosphatidylcholine, enhance the ability of the enterocytes to stay intact. But whether that was the reason for the increased digestibility or is the ability to stimulate chylomicron secretion, we can't really sort that out. And you can see that choline was in greater concentration, so this rumpectin choline is actually showing up in the circulation. And you can see that triacylglycerol in the lymph was also increased. So we think that this might indicate increase in chylomicron triacylglycerol. Okay, so if all of this is true, and it happens in the transition cow, that should benefit production. So a few years ago, we went back to the literature and asked the question, does supplementing rumpectin choline improve productive performance and health of cows? So here's a forest plot from multiple experiments. There, at the time, we found 21 randomized control experiments in which we had a negative control and we had some amount of rumpectin choline from multiple products used across the globe in controlled experiments. There are 66 treatment means, and we use only the data from Paris cows, second or greater lactation. There's not enough data from primipars for us to make sense of the responses. So the way you read this here, each name here represents a reference in the literature. Each row represents a comparison against the negative control. So in the case of Hartwell, it was a factorial arrangement. They had choline and protein in the experiment in Indiana. And then this central line here represents no difference between treated and control. If the point estimate falls to the left, means that the response decreased with feeding choline. If falls to the right, means that the response increased with feeding choline in this case. The gray square represents the weight of that comparison in the statistical analysis, which is listed here. This is a technique called meta-analysis. And the horizontal line represents the 95% confidence interval for that point estimate. So the important points from this is that how repeatable the response is with different managements. And different managements, I mean the different experiments, because they represent different farms, different diets, different everything. Yeah, and there's there's good confidence that the response is somewhat consistent. It falls to the right in most cases. That's good. A single experiment doesn't tell you everything. Okay, but when you have many experiments, that gives you a sense of security in terms of external validity. And in general, the response is positive. That's the overall effect from the mixed model. How positive? Almost 0.4 standard deviations. And this represents about 2.2 kilograms, in this case, of energy-corrected milk. There's an important point here. This here is called I-squared. This denotes whether the response is heterogeneous, meaning that it's not consistent across all the experiments in the data set. So because of that, we explore what is explaining the differences in response between the numerous comparisons here. And there were two things that came up from our mixed models. Okay? One of them, which was good to see, was the dose of choline ion. Okay? The more people fed, the greater the response. So the circles that fall on the 0 grams are our controls for every experiment. The dashed line connects multiple treatments within the same experiment. Okay? If you see a continuous line, it means that there was a statistical significance. So choline either cause a linear or a quadratic change in the response. In this case, I'm showing milk yield. But let's look at energy-corrected milk, which is sort of the standard in the US. It's a milk standardized for 3.5% fat and about 3.2% true protein. Okay? So it's about 0.7 megacalories per kilogram of milk. And you can see here that there is an increase as the dose increase. Our range of doses were from 5 to 25 grams. But you can see the vast majority of the experiments were within this range here, 12 to 15 grams. That's where we are more precise. But those effects were always linear. We never picked up a quadratic response. The second point is that you see there is also an interaction between choline and the metabolized methionine content of the postpartum diet. So what we did is, in the experiments, people described what their diets were. And if it was not very well described, we contact them. And then we put every single diet into the 2001 NRC, so we can have the same platform used to estimate energy, metabolized amino acids, etc. Okay? So then we ask, is metabolized methionine linked to the response of choline? And in fact, it was. Okay? So here is the supply from the multiple experiments of metabolized methionine, okay, postpartum. Okay, this depends obviously on the diet and on the observed dry matter intake of the cows. The more they ate, so experiments that measure intake longer, intake's going to go up. Experiments that last only 21 days postpartum, intake would be less. So the estimated supply of methionine would be less just because intake was less. And then it depends on the composition of the diet. So some diets have very high supply of methionine because they fed room-protected methionine in large quantities, or because those cows ate more dry matter. But if you look as a percentage of the metabolized protein, now the intake doesn't change much this thing. You can see that most of those diets, okay, were between here 1.9 to 2.3% of the MP as methionine. Okay, just keep that in mind. And some had very, very high. They fed a lot of room-protected methionine to those cows, like 20-some grams in the postpartum period. So what did we find? We found this here. Okay? So when metabolized methionine was very low, so here is the control treatment. Let's go to energy-corrected milk, because it's the same thing. But so here's the control treatment, 0 grams of choline ion. Here is the almost 25 grams of choline ion. So when we have very low metabolized methionine, we have a very large response to room-protected choline. When we have a very large dietary metabolized methionine, we still have a response to choline, but it becomes smaller. Okay? which makes biological sense in my mind. It was good to see that mathematically this came up. But we still have a response to choline. So for example, if I go from 0 grams, where I have most of the data, if I go from 0 grams to 12 point some grams, I still get a 2 kilogram response in a diet with moderate to high methionine. This is based on the NRC 2001. If you use a different software, your predictions will be slightly different. And I think Clay can correct me, or you guys can correct me. If you use programs such as the Cornell Biology, which is NDS, EMTS, uses it, this prediction from the NRC would actually be greater, correctly? Yeah. So don't get caught up in, oh, this 225 or this, because different softwares. If you use your own, probably you have a different prediction in the supply of metabolized amino acids. The point here is that you do get responses, but the response becomes smaller depending on supply of metabolized methionine. So what happened to feed efficiency? It went up. So supplementing room protect choline increased energy correct milk per kilogram of dry matter consumed by about 60 grams. So cows became more feed efficient for potentially different reasons. What happened to health? Not every experiment reports health, and that's a problem in literature because most individually fed cow experiments don't have power to measure disease incidence. So it's not uncommon for reviewers to say, just drop this from the paper. Don't report that because your experiment was not designed. When this happens, then we cannot go and assess that using meta-analytical methods. So you can see here, I started with 21 experiments that reported milk production, but there's only 9 to 11 that report health data. So it's a less robust data set. Nevertheless, we still picked up some positive effects. So a tendency for reduction in retained placenta, a tendency for reduction in mastitis in those cows. Okay? So going in, at least in the positive direction, although the data is less robust. So what about after a stop supplementing choline? Because most experiments stop when the cow leaves the fresh pen, so about 3 weeks postpartum. So here are 2 experiments from the University of Florida. Okay, so we fed from, in this here, 255 days of gestation to 21 days postpartum. This here from 250 days of gestation to 21 days postpartum. You can see that the response continued for many weeks afterwards. So in the first one, 2 extra kilograms for 40 weeks. The second one, 2 kilograms for another 25 weeks. And Barry Bradford just replicate this and showed exactly the same. So the response continues. Okay? We don't necessarily know why. There may be multiple mechanisms. I'll give you my take on this. So here's the phosphatidylcholine concentrations in blood around transition, slowest. Okay? In the mammary gland expresses this enzyme here, choline kinase, that phosphorylates choline to make phosphocholine. In human medicine, choline kinase is used as a marker for breast cancer for humans. Okay? So there's a possibility, because choline kinase activation is linked to mitotic division of cells. There's a possibility that this increased persistence of production that went on might be linked to improvements in animal health. That's a possibility. But there's also a possibility that this may be linked to proliferation of mammary cells in early lactation. Okay? So if maybe substrate supply affects the activity of choline kinase, that may enhance epithelial cell proliferation in early lactation that leads to persistent increase in production past the period of supplementation. We just don't know whether that's true. That needs to be demonstrated. But it's just a hypothesis at this point. Lastly, what about body condition score in response to choline? So we mined our data from 2 experiments. There were about 230 cows in those 2 experiments. 223 cows, I believe. So here's the distribution of body condition score of the cows in these 2 experiments in the control and choline when they enter the prepartum period. So it would be your cows moving to the close-up pen. So we had cows with moderate to low body score. We had overconditioned cows. Okay, so excess body fat and more adequate body condition score. So we asked the question, is there an interaction between body condition score and choline on the responses that we measure? So I'll show you energy-corrected milk. Let's focus on this figure here just for the sake, because it's exactly the same. So here I have the effect of room-protected choline was significant. As I move from the blue to the orange, which is controlled to choline, I capture an additional 2 kilograms of energy-corrected milk. But you can see that this effect is independent on body condition score. Regardless of cows having moderate to low or excessive body score, we see exactly the same response. We do not think that cows that respond to choline are fat cows only. We think that skinny cows or low body score cows respond to choline. I think it's a mistake in the industry, it's a misconception just because of the mechanism of action. Most people think that only fat cows respond to that. So lastly, just to capture all this information, we have a lot of data on gene and protein expression in liver. And we sort of built a story which needs to be demonstrated individually with particular experiments. But when cows calved, they mobilized tissue as an adaptation to supply nutrients to the mammary gland. So some of those lipids that are coming from the arterial supply through the hepatic artery will get to the liver tissue, and they are transported by particular transporters in the cell. Supplementing choline does not change the expression of those transporters, at least to the best that we can measure. We haven't seen CD36 fatty acid transfer protein genes to be changed. So we think the entry of fatty acids to the cell doesn't change whether you feed choline or not. Okay? But then when choline enters the cell, it can be direct to, or when fatty acids, enter the cell, it can be direct to different organelles. So it can be oxidized in the mitochondria to generate ATP to the liver. If you have very long chain fatty acids, 20, 22, 24 carbons, it might be direct to this other organelle, peroxisomes, that chops the fatty acid into smaller chains of carbon and then redirects that back to the mitochondria. And in this process, this generates reactive oxygen species. It damages the hepatocyte. Okay? So choline has this, as phosphatidylcholine, this ability to reduce inflammatory response. And phosphatidylcholine alters the fluidity of membrane. So it's possible that the transfer of fatty acids to organelles is affected by supplementing choline. Then when in excess, those fatty acids are re-esterified to triacylglycerols, so the lipid droplets that accumulate in the cell. From the human literature, we know that this is not just the storage of lipids. These are quite dynamic organelles within the hepatocyte. Okay? And the way they are eliminated is either by export or degradation. So they undergo degradation in what's called in phagolysosomes. So we have shown that at least genes linked to formation of the phagolysosome in hepatocytes are upregulated when we feed choline. So we think that the ability of cells to degrade the lipid droplets might be enhanced. I showed to you that in vivo, we increase the rate of nascent triacylglycerol secreted by the liver, which is mediated by synthesis of very low density lipoprotein. I didn't show you, but we have data showing that we and the Dutch have data showing that supplementing room protect choline upregulate genes involved in VLDL assembly and secretion. So it would be 48 microsomal transfer protein. I showed you data indicating that choline increases glycogen content in the liver, either because it reduces glycogenolysis or increases glycogenesis. Our best interpretation of the data today is that it increases glycogenesis. Choline, there's lots of data in literature, and I could show you here in cattle as well, that choline reduces inflammation. So it knocks down inflammatory response, okay, in different models with calves or with monogastrics. So we think this reduces inflammation at the level of the hepatic tissue. I gave you a hint that choline increases fat absorption in the intestine. We don't know the exact mechanism, but it presumably may be because we enhance chylomicron synthesis and secretion, or perhaps we improve the architecture of the intestine such that absorption is enhanced. And lastly, I left you with this hypothesis that maybe choline is affecting genes involved in mitosis in the mammary epithelial cells. So this needs to be proven, but it's possible that the increased persistence of lactation, or persistence of response, I should say, may be linked to proliferation of mammary epithelial cells. So there are multiple mechanisms above and beyond the classic reduction in fatty liver. So just to conclude, in my interpretation of the literature, I think it's unquestionable that choline fits the criteria of required nutrient. Not an essential, it's different. Ruminants can synthesize that, tissues can synthesize phosphatidylcholine or recycle it, although we truly have no data. To the best of my knowledge, I contact maybe 30 different people who work only on choline, and nobody can give me a straight answer whether there is de novo synthesis of choline molecule by the cells in mammals. They all tell me it is essential because there is no synthesis, but they cannot point that to me. But obviously there's recycling of phosphatidylcholine. Supplementation reduces triacylglycerol in the hepatic tissue. I didn't show you the intake data, but there's a slight increase from the meta-analysis in intake data. Very hard to pick up with individual experiments. So I would probably ignore this, but it does increase consistently energy-corrected milk, and that's a good thing, and tends to reduce the risk of some disease. The response to RPC is influenced by the supply of metabolized methionine. Okay? It's influenced by the amount of choline ion that we feed, so those two things are important. The benefits are observed regardless of body condition score of cows, and that response extends beyond your period of supplementation in the early lactation group. The mechanisms are probably multiple. We simplify the story because it's the easiest to tell, is the fatty liver story, okay? But there's probably changes in animal health that explain some of the response. There's reduction in inflammatory response that probably consumes less nutrients and secures partition of nutrients to the mammary gland. And there are other non-classical mechanisms, such as perhaps mammary cell proliferation, etc. So thank you for your attention, and hopefully we'll have some discussion later on. So thank you.